Good morning, everyone. I'm delighted to welcome you all today. Today, we're going to explore an important topic in operations and customer service, hold time studies. This presentation is titled Hold Time Studies and is presented by Dr. S.K. Singh, Ph.D. Over the next few minutes, we'll discuss what hold time studies are, why they matter, and how they can help improve service efficiency. So, let's get started and dive into this interesting subject. This slide highlights the importance of ensuring that materials stay within their approved specifications throughout the hold period, in strict accordance with GMP requirements. A hold time study is a crucial part of this process. It's a documented, scientific evaluation that helps us determine the maximum amount of time in process materials, intermediates, bulk products, or even cleaned equipment can be held under specific conditions without compromising their quality, safety, efficacy, or compliance. The goal is to make sure that before moving to the next process step or final packaging, these materials still meet all quality standards. Manufacturers have a responsibility to ensure their products are safe and effective, which means using validated processes and adhering to clearly defined procedures. GMP emphasizes proper storage to prevent any impact on quality, and that's why scientifically justified maximum hold times, supported by relevant data, are so vital. Ultimately, this ensures that every product leaving our facilities maintains the high standards our patients expect and deserve. Let's take a closer look at the scope of these guidelines. They focus on key considerations for designing hold time studies, specifically for non-sterile solid dosage forms. However, many of the principles we discuss are also applicable to other types of dosage forms. The main goal here is to provide general guidance for both manufacturers and GMP inspectors. It's essential to emphasize the importance of using scientific data to justify hold times. This helps ensure that materials, intermediates, bulk products, and finished products consistently meet quality and acceptance criteria. Overall, these guidelines are about establishing a solid, science-based foundation to maintain product quality throughout the manufacturing process. Hold time is essentially the designated period during which raw materials, intermediates, and bulk products can be stored under specific conditions and still meet quality standards. It's a crucial aspect of manufacturing because it helps us ensure that products stay within their specifications throughout the process. When we talk about hold time studies, we're setting clear time limits at each stage of production. This way, we can verify that the product maintains its quality from start to finish. These limits are usually established before the product hits the market and are confirmed during validation to make sure everything is consistent and reliable. Identifying the critical stages in the process is key. We often do this through risk assessments or flow charts, which help us pinpoint where things could potentially go wrong. From material preparation all the way to final packaging, these are the steps we focus on to ensure quality control. Overall, understanding and managing hold times is vital to maintaining product integrity and meeting regulatory requirements. It's all about controlling the process so that we deliver safe, effective products to our customers. Let's walk through an example of the manufacturing process for coated oral tablets. Starting with binder preparation and granulation, here we need to consider the quality and consistency of the granulate itself. Moving on to wet granulation and drying, the focus shifts to ensuring the dried granulate maintains the desired properties for further processing. Next, when we go from dried granules to lubrication and blending, it's important to consider how well the lubricant is distributed within the blend to facilitate compression and coating steps. Speaking of compression, this is where we form the tablet cores, which are then prepared for coating. When we consider the coating solution, the key is to ensure the coating material is prepared correctly to achieve the desired film properties. After coating, the next step is packing, either in bulk form or individual packaging, so we need to keep in mind the condition of the bulk-coated tablets. Finally, the process concludes with packing the bulk into finished dosage forms, ready for distribution. Throughout each of these stages, The overarching theme is maintaining quality and consistency, which is fundamental to meeting pharmaceutical standards and ensuring patient safety. Let's now look at the aspects to be considered. When we're discussing this topic, it's crucial to understand the key points thoroughly, but in a way that feels conversational and engaging. First, we want to highlight the most important concepts, because these form the foundation of the overall message. For example, understanding the core principles helps us see the bigger picture clearly. It's also essential to connect these points to the overall theme of the presentation. This helps reinforce how each aspect fits into the larger context, making the information more meaningful. Remember, considering these aspects isn't just about ticking boxes. It's about really understanding how they influence our approach and decisions. By keeping these points in mind, we ensure our analysis is comprehensive and aligned with our objectives. So, in summary, paying attention to these key aspects allows us to make informed, thoughtful decisions that support the overall success of our project or initiative. When conducting hold time studies, it's essential to have a written protocol in place. This protocol should clearly define the activities involved, the test parameters we need to set, the acceptance criteria, and how we'll report the results. Having these details documented helps ensure consistency and reliability in our testing process. It's also crucial that the testing uses validated, stability-indicating methods. These methods are specifically designed to accurately measure the stability of the product over time. If microbiological aspects are relevant, they must be considered to ensure the product remains safe and effective. Additionally, we should apply a risk-based approach when selecting batches for testing. This means choosing batches that represent the highest potential risk to ensure our results are meaningful and protective of quality. When storing samples, it's important to keep them in representative containers, those that mimic actual packaging, to reflect real-world conditions. We need to test samples stored under worst-case scenarios as well as typical production conditions. Comparing these results against baseline data allows us to confirm that the material remains within established control limits, ensuring ongoing product quality throughout its shelf life. Let's take a moment to consider the key aspects we need to keep in mind. First, it's important to thoroughly understand each aspect, but we also want to keep the conversation engaging and approachable. When discussing these points, I'll highlight the most critical concepts, making sure they stand out so they're easy to remember. Connecting these points back to our overall theme is essential, as it helps us see the bigger picture and how each aspect fits into our goals. By doing this, we ensure our discussion is not only informative, but also meaningful and relevant to the broader context. This approach will help us make smarter decisions and implement more effective strategies moving forward. Let's take a moment to look at the reference on this slide. It points us toward the supplementary guidelines on good manufacturing practice, specifically focusing on validation and non-sterile process validation. These guidelines are part of the WHO Expert Committee on Specifications for Pharmaceutical Preparations, which is a key resource for ensuring quality in pharmaceutical manufacturing. The document was published in 2015 as Annex 3 of the WHO Technical Report Series, number 992. What's important here is that these guidelines help us understand the expectations for validating manufacturing processes, which is critical for ensuring consistent product quality. Validation, in this context, means providing documented evidence that processes are capable of consistently producing a product that meets its quality specifications. When we talk about non-sterile process validation, it's especially important because it helps us identify and control variables that could affect the safety and efficacy of the product, even in non-sterile environments. This reference serves as a foundational document, reinforcing the importance of rigorous validation practices as part of good manufacturing practices overall. In essence, these guidelines support our overall theme of maintaining high quality and safety standards in pharmaceutical production, ensuring that every batch meets the necessary specifications for patient safety. For further support, please feel free to contact us. We're here to help you with any questions or additional information you might need. Thank you so much for your time and attention throughout this presentation. Remember, we're committed to supporting you every step of the way as you apply what you've learned today. Don't hesitate to reach out. Your success is our priority.